Physiology is the study of processes and functions in living organisms. The ESTECK Complex is a system for the estimation of the internal environment and main regulatory mechanisms of the homeostasis, with mathematical and standardized interpretation. The American physiologist Walter Cannon introduced the concept of homeostasis and the existence of steady conditions of regulatory mechanisms. Changes in cell function always occur in the context of a whole organism, and different tissues and organs affect one another. An important part of physiology is understanding how different parts of the body are controlled, how they interact, and how they adapt to changing conditions. From Lippincott, Medical Physiology. A disease or death is often the result of dysfunction of internal environment and regulatory mechanisms. Understanding the body processes, responses and functions is clearly fundamental to the intelligent practice of medicine. The ESTECK is a non-invasive medical device using tactile electrodes and biosensors, and allows the data recording of 5 technologies in only 5 minutes. The recording are displayed in real time, and then analyzed and saved in the software. The system is a combination of technologies. The EIS technology, bioimpedance low frequency and in bipolar mode, provides the tissue density, mitochondrial activity, and tissue perfusion, according to the electrical properties of tissue. The spectrometry technology using oximeter provides the autonomic nervous system activity, SPO 2%, and hemodynamic indicators. The bioimpedance analysis in tetrapolar mode and frequency of 50 kHz provides the estimated body composition parameters. Due processing analysis for the spectrometry technology. The pulse oximeter displays the SPO 2%, vascular waveform, and heart rate. The photoelectrical plethysmography is the signal processing analysis of the waveform output of the oximeter, and provides hemodynamic indicators. The heart rate variability is the signal processing analysis of the time variation of the heart rate, and provide indicators about the autonomic nervous system activity. Due processing analysis for the EIS technology, measurement of 22 segments of the human body in bipolar mode. Site of investigation according to the Venn diagram of the used frequency of 700 Hz. Original signal transmitted to the software. Signal processing analysis. Step 1. ESG graph, real conductivity of the 22 measured segments. Step 2. Calculation of the cell's metabolic needs, value A1. Step 3. Second derivative ESG from the ESG graph and value A1, and calculation of the parameter SDC, related to the segmental interstitial fluid sodium concentration, and segmental ATP pump activity. Step 4. Dynamic processing analysis, and calculation of the parameter EPA-SPA, related to the current distribution, and segmental tissue density, parameter alpha of the Cole equation. Step 5. Spectral analysis of the original signal fast Fourier transformation, and calculation of the parameter HF-VLF, related to the general interstitial to the blood viscosity. Step 6. Cross-analysis of the segmental parameters SDC, EPA-SPA, and HF-VLF, for the estimated tissue perfusion. BIA analysis following the sending of weak intensity at a model frequency of 50 kHz through active tactile electrodes. The BIA sensors measure the resistance and the reactance between 2 other passive tactile electrodes. Tetrapolar mode measurement, and provides estimated body composition parameters. Results of the ESTECK system. The results are displayed in table form with the value, scale, normal range and units of each indicators. The table displays the SPO 2%, the original wave PTG, and mathematical acceleration of the original wave ST PTG. The analysis of PTG and ST-PTG provide hemodynamic indicators as cardiac output, stroke volume, blood volume, ejection fraction. The table display the autonomic nervous system indicators. The main indicators are MSSD, indicator of parasympathetic activity; MXDMN, indicator of the stability of the heart rhythm; HF, indicator of parasympathetic activity; LF, indicator of the sympathetic system activity. LF-HF ratio considered by some investigators to mirror the sympathetic-parasympathetic balance. EIS table. Segmental analysis of the living tissue or display, with the two parameters STC mitochondrial activity and EPA-SPA tissue density. The BIA results are displayed in three parts: general distribution body composition analysis and the obesity evaluation. The body composition analysis is including the fat-free mass, the fat mass, the total body water, the extracellular water and the intracellular water. ESTECK outcomes: non-invasive, quick, usability, and low cost. For the patient, understanding the treatment and goal of the treatment, compliance. For the practitioner, organization, save time, 4 measurements in 4 minutes, results immediately available, follow-up of treatments, corrective or preventive actions that the patient can understand, referrals, income increased. Thank you for your time.